to everyone, and to welcome everyone to today's lecture titled Microbiome Functionality as a Signature of the Environmental Conditions. It is my great pleasure to introduce our speaker to you today, Professor Jana Bromberg. She's a professor at the Department of Biochemistry and Microbiology and the Department of Genetics at Rutgers University, New Jersey. Her research goal is to understand how function is encoded in genetic data on different levels, such as single gene, a genome, and metagenome, and her lab at Rutgers University using bioinformatics to predict protein function and analyze genome variations. Some of the project the lab is working on are developing an in silico mutagenesis methodology, which will define functionally important residues in protein sequences, and analyzing the effects of genomic SNPs on the overall organism fitness. Professor Bromberg, thank you so much for being here with us today again, and the floor is yours.
Thank you so much for that introduction. I haven't heard myself introduced in a while due to wonderful COVID restrictions. It's a pleasure. So I didn't actually know what to expect from you guys, as in what you would know and what you wouldn't know. So the first thing that I want to do is say, if you are not comfortable with what I'm saying, just interrupt me. And since I'm going to be presenting, I'm not going to see chat. So just speak up, turn on your microphone and speak up. All right, I'm going to share my screen. For some reason, I can't. That is interesting. Okay, here we go. Is everybody seeing this?
Yes.
Okay, cool. So let's get started. First things first. So a while back, I used to attend physics talks, and this was one of the best things that I've seen there. So normally when you start out your talk, you start out with about 100% understanding in the room. So everyone that's listening to the talk is just on the same page. And that understanding drops off a little later. Now, in an ideal situation, what happens is that the understanding of the material never drops below 50%. And that should happen somewhere halfway through the talk. And then by the end of the talk, people should be able to tie things up and figure it out and come away with at least 75% of the things that have been talked about. Now, this is what the typical presentation looks like, right? That means that by the time the talk gets into any detail, we have lost everybody, and that's not something that I'm looking forward to having. There's no point for me to talk if you're not going to understand what I'm saying. So if there is any issue, if anything is unclear, do speak up and stop me in the middle of what I'm talking about so that you can actually keep on track with what's going on. All right, so let's get started. First things first, microbes are everywhere. Now, you guys probably know this. Microbes exist in the gut, and on the surface and the skin of animals. They're in the ocean, in the sea, they're in the soil, they're everywhere. So microbes tend to occupy whatever niche they want to, right, or they could occupy. And one of the studies that's becoming very common right now is figuring out what microbes are where and what are they doing. So we do this via microbiome sampling. And the problem with microbiome sampling is basically that it's really hard to figure out what's actually going on within a sample that you've collected. So let's, for argument's sake, say that you have bacterial genomes that are present in the sample. So what we do with sequencing is we will cut this up. So there is no PCR usually that's applied to microbiomes, because you don't want to augment anymore the primary genomes that are there. So you don't want to increase the number of blue, but by applying PCR, for instance, you're not going to get at random the reds, the greens over there. So what we usually do is we just take the metagenome from the sample directly. We cut the genomes into small fragments, and then we sequence them using regular sequencing techniques, and then try to assemble back the genomes that we've cut up. Now, what that would ideally look like is like this. You would have a lot of the blue and some of the green and some of the orange and the red. Of course, as sequencing is more or less random, you're not going to get that. So what happens? Well, I usually like to draw this kind of analogy. So let's say you have this wonderful library, the Trinity College Library in London, in Dublin, and I ask you to tell me what is in this library without actually opening any of the books. Right? You can do other things to the books, but you can't read the title or whatnot. So the way that microbiome analysis, metagenomic analysis currently works is you would go in and shred every single book in this library, right? Now, that's already a problem because you're missing a whole bunch of things that you've just, destroyed by shredding the books. But what you will do then is scan these individual shredded pieces, tiny little pieces of sequence. And what you're going to do is try to assemble those little tiny pieces of sequence into books. And by the end of this thing, what you're going to get is 2 books, probably the most common ones in the library. So hopefully that makes sense to everybody. So just simply taking the data out of metagenome sequencing and trying to figure out who is there on the basis of assembled genomes is exceedingly difficult. So what people do instead is they try to recognize what bacteria are there on the basis of the individual reads. So they will take this read and they will align it to the known, or every read, and they will align it to the known bacterial genomes to try to say this read comes from this organism and or it comes from this gene. Now, we at the Bromberg lab felt that the reference genes or genomes are not important. And what is important is the function or the molecular function that these genes would have carried out in the complete organism. But we now only have these pieces, so how do we know where these pieces belong? So for this purpose, we built a method called Mi-faser. We took the existing reference genomes in these red and blue over here circles, and we extracted from them only the genes that have been manually, experimentally annotated as having a particular molecular functionality. And this molecular functionality is highlighted over here by this, what is called the enzyme commission number. So that's 4 digits separated by dots over here. So if 2 proteins have the same 4 class EC number, so like this one in red that comes from one organism, and this one in blue that comes from another, they have the same EC number assigned to them, which basically means that the genes in these 2 different organisms are carrying out the same type of molecular function. So we got these reference proteins out, and then we cut them up into little tiny pieces, which you would get from sequencing. And then we translated those pieces. This is from genome data, right? So basically the corresponding genes, and then everything else that's in a genome. So some things will not be annotated. But we translated all these individual pieces into a protein state. So that's a 6-frame translation. And then we align these pieces to the proteins that we originally had in the data set, making sure that we don't align blue to blue and red to red, because those would be the same thing, right? So basically reads coming from the same gene. Now, for every alignment, we had to figure out whether this is an alignment that reflects the same functionality or different functionality. So here, for instance, this 1.1.2.1 in blue is aligned to the read that comes from the gene in the red gene that had the same functionality as this one. And so we assigned to that a correct, this little blue circle over here. Now, on the other hand, if you look at this alignment, since these numbers are different, what you're saying is that this alignment is incorrect and should not have happened. And we can do this for everything in our list. Now, once we do this, we can place these dots into a two-dimensional space of sequence identity versus alignment length. Now, you see in this plot very clearly that the green dots happen to be over here in the upper right corner, while the red dots tend to be on the lower left corner. And that's to be expected of the alignment game. So what we did is we then, using our data, optimized a curve. This is a gradient descent curve that separates the space into the red and blue and allows us to say that any alignment that is above this curve, right? And this is the formula that we had optimized. So sequence identity minus 352 length to the power of blah, right? So it's a gradient descent curve that's dependent on the length of the alignment, right? But the idea is that everything that's above the curve is a correct alignment representing a read that has a particular function in our data set, versus anything below the curve. So like, right, these red dots over here is incorrect alignment. And if what we found is that this way of doing things, so that's indicated in this black line over here, which highlights the precision of the method versus how many sequences can be annotated in this way, the recall, right? This type of the way of doing things was a lot more precise than what normally people use. So things like BLAST, for instance. So at, let's say, faser equal 20, so this is something that's 20 points above the curve that we had just optimized, we were able to predict 50% of the reads with 90% precision. And now remember, our annotation is actually very, very good in the sense that it's manually curated and there is experimental evidence for it. So that was a really interesting result. So our performance was significantly better than the state of the art. And then we created the website for it. So the way that we had programmed it, this method Mi-faser, is actually extremely fast. So you can submit your metagenomic data if you have any, and it will generate the results for you. I tell you what functional activities are being performed by the organisms in your microbiome. And so this is all very cool. It's very fast and very accurate, but there is one problem. So this precision comes at a very large price. So if you look at the database of all known proteins that have manual curation and manual annotation of their functionality, these are these Swiss prot proteins. There is about 550,000 of them. And actually, curiously, there were about 550,000 and there are now 580,000 in the 2 years that had passed since. The reason why this growth is so slow is because people have to actually curate manually. So you can run anything you want in a computer, but if you had to actually do the experiment, that's problematic. So there aren't that many proteins in the world that are annotated manually. But even of the ones that are, only about 350,000, so about half, are bacterial. And of those, only 18,000, that's the light blue thing over here, actually have evidence of protein existence. That is, somebody not only translated it from the genome data, but also did some sort of analysis, for example, mass spec or something else, that tells you that the protein not only is encoded in the genome, but also exists. And that's only 18,000 proteins over here. And of these, a tiny fraction has also been validated for the functionality that it has. So somebody had evaluated the biochemical activity of this particular protein. So the grand total of proteins that we were looking at was 2,800. And that represented about 1,200 activities, so molecular functions, enzyme commission numbers. And if you take that as a reference, which is a tiny, tiny little set, but if you have this super precise method for evaluating functionality, then all you're able to annotate is about 2% of any given metagenome. Now, this is a really small number. Arguably, this is a very important piece of the genome, right? So this is of the metagenome. This is the enzymatic activity, or a lot of the enzymatic activity that is being carried out by the microorganisms, but it's still only 2% of the metagenome on average. So what does that mean? I'm going to come back to this a little later in the talk, but for now, let's just assume that you can represent every metagenome as a vector of functions that this microbiome may be carrying out, right? So you have microbiome 1 over here, and it has a small amount, like blue means low amount, of function 1. It does have a higher amount of function 2, and you can do this in coding for every single microbiome that you're interested in. And then you can ask a question: how similar are microbiome 1 and microbiome 2? And one way to figure this out is to ask, what is the Euclidean distance in this m-dimensional space of functionality between these microbiomes, right? And this is going to be important. So let's have an example. What we have here, for instance, is this DeepHorizon Oil spill. Some of you may remember this. This is now a long time ago. So I think it's over 10 years now ago that this happened. But we had the oil spill happen over here, and the oil spilled and moved around the Gulf of Mexico in the US and landed on these coastlines, right? So there was oil all over the coast, and the beaches had oil contamination. So scientists had gathered the beach data, the sand data from the contaminated sites because they had a reference point. They had sampled these beaches previously when it was still not contaminated. So now we have these microbiomes sampled from these beaches at different points in time. And if we look at them, we have oil points, we have post-oil points, so after the oil has been cleaned up, and the pre-oil. So this is the reference point where they had the microbiome data. So we've gathered this microbiome data, and now we can compute the Euclidean distances between the individual samples. And what you see here is that if I ask for the similarity, this heat map represents the similarity between the individual microbiomes that have been gathered at different times. So yellow means there is low similarity between rows and columns, and rows and columns are these samples. And red means high similarity. And what you see very clearly, because you can build a similarity tree on top of this, is that the pre-oil samples all look a lot more like each other than they do like post-oil and oil samples, right? So that's expected because these samples did not experience this shock to the system of having a lot of oil being spilled. On the other hand, the post-oil and oil spills samples look more similar. So if you take this sample over here, for example, the OSH, that's an oil sample, but it actually kind of looks like the post-oil samples. So that means that even though the oil has been cleaned up, that is, the oil spill samples look a lot more similar, it hasn't been completely cleaned up, or the environment hasn't completely yet recovered. And so another way of looking at it is rather than using Euclidean distance of the entire vector of functionality, what we can do is we can reduce the dimensionality space from these 1,200 enzymes that we were looking at to the, let's say, 2 dimensions using this method called non-metric multidimensional scaling. You could also use other methods like TSNE and UMAP, and even principal component analysis, but this is the method that is most applicable in our scenario. So what you see here is that these dots over here, the triangles on this side represent the pre-oil spill microbiomes. These empty squares are the post-oil microbiome, and the filled samples are the oil microbiomes. And these are very different, but you could almost imagine this sort of a scenario where you're sort of going from this oil spill catastrophe through some cleanup over here and into the kind of microbiome that you would have had pre-oil. And you could infer all of this only using the enzymatic activity, which is nice to be able to do, because you can sort of say, well, not all of the activity of the microbiome is necessary to be evaluated. You could actually highlight the particular molecular functions that are important to the recovery of the environment. Okay, so that was the Gulf Beach. The other thing that we did actually a lot more recently is sampling the spring snow, not ice, in Svalbard. Svalbard is a town in the Arctic region of the world, where they say there is more polar bears than people as a population in that town. But our collaborators had gone over there to do their sampling, and what they did is they sampled early in the spring, so early March, and they also sampled the snow towards later spring, so towards May. And we did the same type of analysis as we did with the Gulf Beach, but here we used both the DNA and the RNA from the early samples on the left and the late samples on the right. And what you very clearly see is that there is a difference in the way that these distribute. So the early samples are actually all over the place. And of course, you don't necessarily expect DNA and RNA to mirror each other, because RNA just concerns a very specific portion of the molecular activities. But you did expect, or at least I did expect, that some of the DNA samples were going to group closer together, kind of like what we see in the late spring. So early spring, samples all over the place. Late spring, they're closer together. And one possible explanation that we had for this was that all of these samples that we were looking at in the early spring was really about not actually a live bacteria, but if you gather a sample from the snow, you're also picking up the DNA that would have stayed there from last season. Now, because it's so cold in the winter, the DNA doesn't really degrade very much. So we are able to sequence it here. So what you're seeing here is basically the remnants of bacteria that would have been alive the summer before. Whereas over here, what you're seeing is probably a very concerted community that's growing, right? So that's why you can measure samples in different places and they're going to be kind of similar. And we thought that this was actually a very good illustration of what happens over the development from early spring to late spring. Another thing that we observed here is that our collaborators measured oxalate, acetate, and formate concentrations, which are these organic acids that are usually either deposited from plant growth or are produced or deposited from abiotic sources from any activity. But because in Svalbard, abiotic sources are limited, we sort of assume that increase in these organic acid concentrations actually refer to the increase in growth of the live stuff, right? And what we see here is that the oxalate and acetate and formate concentrations actually correlated very highly, well, mostly highly, with these biosynthesis, fatty acid biosynthesis pathways, which we were able to infer from the molecular composition of the metagenomes that we had sequenced. And what that means is that so these pathways, upregulation of these pathways indicates that bacteria are growing. And that correlation with the organic acid suggests that these bacteria are growing and they're either eating or producing these organic acids, right? And given the organic acids are more of a plant production kind of system, that just means that these bacteria are taking advantage of the fact that the plants are growing and also growing. And what you see here in these last two lines is that you have a positive correlation with geraniol degradation and a negative correlation with styrene degradation, which suggests that these bacteria that are growing are switching from eating geraniol to eating styrene. And this is actually the first time that things like this have been recorded at these really low temperatures. So that's an important finding. But I think I'm using this point only to illustrate that by looking at microbiomes in this way, you are able to infer things that you would otherwise not see. Okay, another thing that we did, and I'm sort of continuing on the same topic, but that you could do these samplings in all kinds of environments. So we did the Gulf Beach, we did the Svalbard, the snow samples. But now what we're going to do is we're going to look at the Prader Willi syndrome, which is a genetic disorder which causes a lot of problems for little kids. One of the biggest problems that they have early on is that they're obese. And the reason they're obese is because they never are satisfied with how much they're eating. So that's a genetic way of not being able to say, okay, I'm satisfied, I'm full, I'm gonna stop. And there are multiple ways of treating these children, but the biggest way right now is the diet, right? But of course, you know, it's, if you've ever tried to be on a diet, it's hard. Kids don't understand what it's like to be on a diet, and these kids are somehow mentally deficient a little bit. And so what we were looking for is, can we see a microbial signature of, you know, possibly satisfaction in these kids, right? Of how are we feeding them? So I'm going to explain in more detail. So there was a study in China where they had these kids had a transplant of microbiome, and they were on a diet that was not limited in the amount, but rather only limited in the types of things that they could be eating, which is a much easier thing to enforce with children, right? So they had a transplant of a microbiome, and they also had a diet. And this is what we observed over here. This is a day 0, and the y-axis here represents the diversity, the functional diversity of the microbiota that was sampled from the fecal samples of these children. So day 0 means this is before any transplant. And what we see here is that functionally the microbiota of all of these kids are actually very similar. So they have the same problem. Right? This is fairly unusual for microbiota in the gut, because we tend to have different microbiota. All of us have our own microbiota depending on the diet, but these children had the same microbiota. Now at day 30, post the transplant, and they're now on the diet, right? So you see that the microbiota diversity, actually the functionality increases, and this star over here represents that it increased significantly. At day 60, it's even more increased, and then day 90, it's somewhat more increased. And what you see is a very clear trend that over time, these children get a very different microbiota, or a somewhat different microbiota, and that this diet/microbiome intervention actually resulted in kids losing weight. Every child lost weight. So this was an interesting observation, that you can actually alter how both the nutrients are absorbed and whether the kids feel full, right, by simply changing their microbiomes. But also in the microbiome, what you see is different ways of dealing with the situation in every child. So that was an interesting thing to look at in kids, that in obese kids. But one thing that I wanted to point out is that it's not very clear that you necessarily will see some signature immediately just by looking at samples. And one example of this is this subway microbiome sampling that has been done for a while. Right, so people have been collecting samples from microbiomes on the subways and sending them in to Cornell over here across the river to sequence and to try to figure out whether the individual microbiomes differ. And one of the things that was done with this type of sampling is we were told that we should be able to predict, using the microbiome data, where this microbiome comes from. So I can tell you that if I look at the data, and I had specifically removed all color schemes, we're looking at Oakland, Hamburg, New York City, Ofa, Porto, and a whole bunch of other places, right? So they're highlighted. I could highlight them according to the city that they were from, but if I look at this mess, there's obviously no clusters, right? So I can't infer which city each one of these came from. So maybe I can draw a line over here, like a diagonal line this way. But there is no obvious distance-based clustering. So if I put the color scheme on it, this is where I know what's going on, right? You can tell me, oh, okay, now that I see it, I can see that there is a cluster. That's true. There is more things that you can do computationally, but even if you were to do this, you see, for instance, New York City, which is a very diverse system and a subway system. Both the city and the subway system are very diverse. But you see New York City samples over here in green, and over here in green, and over here in green, and there is no clustering that would have been able to recognize these. So what do you do in this case? Well, one thing that you could do is you can do machine learning. So you can train a model to recognize nonlinear separation of the individual samples in space on the basis of the functions that you actually have, right? So when we did that, we were able to see, let's say, something that we were able to predict whether something came from Oakland or not, Hamburg or not, New York City or not. Now, the performance of these wasn't necessarily 100%, right? So you could still get errors over here. So if you look at Hamburg, let's say at score of 1, you have a sample that's not from Hamburg, but it's scoring 1, as do the Hamburg samples. So there are still mistakes, but at least it's a lot better than simply looking at the distances. Okay, I hope you guys are still on board with me, because otherwise I feel like I'm talking to myself. Is everybody understanding what's going on?
I hope we are. If somebody has questions, they're free to ask.
Okay, awesome. Thanks, Sophia. All right. So the other thing that we did is we looked at the microbiome versus a genomic relationship. Not directly, but we had a family, right? So this is a family. If the samples are highlighted in a solid line, we had fecal samples from them. And in this family, it appears that there are 2 cases of Crohn's disease. So right here, this person SO9 in red, and this person SO1 in red. And what we see for this family is that SO1 and SO9, so Crohn's disease samples, are very different from everybody else, right? So here's our tree. This is very different samples. But that's easy, right? So for somebody who has Crohn's disease, which is an autoimmune gastrointestinal system disease, there's obviously going to be a microbiome difference, right? But I can tell you even more. So SO1 and SO9, judging by the length of this branch over here, are actually not very similar to each other. So even though both of them have Crohn's disease, they're actually not very similar. However, they are very, very different from everybody who is healthy. And so we wanted to track what happens with these healthy people, right? So the first thing that we observed in this system is these SO7 and SO8, which is this married couple living together, right? They're all the same age, or more or less the same age. So these two, this is a married couple which is more or less the same age. And their microbiome appears to be very clearly similar. Now, the question here becomes, are they eating the same thing and therefore their microbiome is similar because they live in the same house? That could be the situation, right? So we don't know, but we know that they're the same age, more or less, and their diet is similar. They're in the same household. So the next set of people that's similar is SO3 and S12. Now, SO3 is this woman over here, and S12 is this woman over here. They live in different households, but they're both women and they're of the same age, more or less the same age, same generation, right? And that's why it appears that's why their microbiomes may be similar. Of the same type of relationship is this pair over here. Sorry, I forgot about SO6. SO6 is a man of the same generation as SO3 and S12, but he has a slightly different microbiome than them. And we suspect it's because either he's living a separate household, or more likely because he's a guy. And I'll tell you why more likely in a second. So these are two women of the same generation, and this is a man of that same generation. So the next very similar couple is SO5 and S10. And SO5 and S10 are these children over here. So this girl in H1 and this girl in S10, in H3. So they have very similar microbiome. So now the signal sort of confirms itself, right? So women tend to have similar microbiomes, and age actually matters over here, right? So if then I look at SO4, who is this person over here, right? Then this is a boy living in the same household as SO5, but his microbiome is different from SO5 and different from S10, which is what I had been pointing out before. So now the only person that remains here is this S11 person, who is this girl over here. And you may want to ask, why is this girl different from everybody else? And why does she look like this other couple, right? So this grandparents couple over here. So this person, S11, looks like this grandparents couple. I can't answer that question. But what I do know is that S11 is a child, not a fully grown child, but a baby, right? So S11, well, at that point, that she was a baby. So their microbiomes are actually significantly different from adult people and also more grown-up children. So what you see very clearly here is that those people that are related tend to have the same sex and be of the same age. And this is what's happening in this particular plot. So microbiome has a lot of signal for this particular type of differentiation. One other thing that I want to mention is that going back to the Crohn's disease patients, what you could do by this type of functional annotation is try to understand what exactly is causing the disease, for instance, Crohn's disease in this case. And what we asked is, what are the pathways that are upregulated in these people as compared to everybody who is healthy? And what we saw is these two that were the strongest upregulation, right? So one is lipopolysaccharide biosynthesis, which is known to promote inflammation, right? So it's a production of bacteria, but it's known to promote inflammation in hosts. And the next one is glutathione metabolism, which is helping bacteria survive inflammation. So in trying to figure out what are the causes of Crohn's disease, you can consider the fact that it's not just the genetic stuff that's happening, right? It's not just the Crohn's disease is caused by the genetic malformation of the individual, but also you can talk about the fact that there is microbiomes or microbes in the environment, in the gut of this individual, that may be both promoting the inflammation and targeting survival in this promoted state, or highly inflammatory state. So I hope by this point that I've convinced you that looking at functional similarity of things is actually very important. But now I want to get back to this situation that we had at hand. We start out, we're looking at 2% of the data, right? Microbiome analysis using homologous sequences is actually very limited, right? We can't really infer things that we haven't seen before. So we start with 2%. But what if we wanted to get to the star in the sky that's 100% annotation? How would we go about that? And one of the things that we could do is actually thanks to the advances in deep learning. So for those of you who don't actually follow this whole idea of deep learning, there is this notion that you can recognize patterns in a way which was not available via standard machine learning before. And I'm going to go through this very, very fast, and I'm not sure it's going to be very helpful to you, but please feel free to stop me. So the way that we think about problems in biology, in genomics anyway, is by looking at sequence. So we have the sequence ACTTT, and it's a collection of letters that actually encode something. Nobody of us actually speaks the language of genes, right? So this doesn't make sense to us as a word. But the language of genes exists, just like English language or Serbian right? So you would have a language that you need to understand before you make any statements that you could make sense of, right? So to read a book, you should first learn the language. But before you even learn the words, you need to be able to understand what these words are. So in deep learning, at least in genetic version of it, what we try to do is we try to look at to make the machine learn the language before we even ask the question, right? So our main question is, what is the function that it's encoded by these reads? But we first need to make the machine learn the language of the reads. And one way to do this is to say, okay, can I, using the existing letters, predict what the next letter would be? So for instance, here, can I, using this sequence over here up to the G, predict what's covered by this particular blue thing? And then they can move one over and I can say, now using this sequence, can I predict the next thing? And I can do that consistently over a fairly large amount of time. Now I can tell you, if the more I do it, the better I get. So the performance improves as you go to the right of this plot. This is the epochs of training the machine learning, but we don't really get very high, right? So without knowing anything else, just the letters, right, you could also only get that good. However, at 40% accuracy, we are actually a lot better than random, right? So if you think about the four possible nucleotides that you could have, picking one at random would give you 25%. We're at 40% accuracy. And we get there faster if the data that we feed to the neural network is more diverse. So if we take organisms that are different by class, we get there faster than if we just randomly sample organisms, no matter how many of these organisms we take. And that's an important point, because if you're trying to teach a machine to recognize a language, that's going to take some time. So the less data you take, the better, the faster it will train. But you want to make sure that your data is actually fairly diverse. So once you've made the model learn the language, you can train it for a downstream task. So once it knows the language, you can ask it what's going on with a particular type of analysis, right? So what happens is this: we have this input of sequences, and first we try to predict the next letter. What that does is generates this nonlinear, very weird multi-hyperplanar embedding, right? That actually tells you about the next letter. But then you could ask the next question. You could say, rather than what is the next letter, can I use the language that I've learned here to make a prediction of the annotation of Mi-faser? Right, so if Mi-faser annotates functionality, then the prediction of Mi-faser annotations should also be accurate. And in fact, we can. So this is now without alignments, just using the sequence that we've looked at. We can predict that EC number, right? The first digit, for instance, with 90% accuracy here, 80% accuracy there. So we're using this model to predict the Mi-faser prediction. That's a double prediction there. And in fact, we do really well at every digit. So first digit, second digit, third digit, we still do well, right? But here's an important point. If we wanted to skip out from this Mi-faser world where we're limited by the number of functions that we know, we have to be predicting not Mi-faser annotations, but rather predicting the functions of the individual proteins without alignment, without homology to anything known. So one major point to remember is that reads from homologous sequences, or that is sequences that are similar, are not necessarily going to be similar themselves. So if I look at the tetracycline repressor protein and I limit my world to actinobacteria and firmicutes, what I see is that there is a whole bunch, I don't know, 12 or so different orthologs or similar proteins across the whole community of bacteria. So what I expect them to be is sequence similar on a large scale. But when I look at them in detail, I see that, for instance, this 229 protein aligns to another protein of 219 residues only over 15 15 amino acids. And what you see here is that even two orthologs could have limited shared sequence. But the problem is more than that. If I take a read that covers that short sequence that's homologous, then I'm okay. But if I take a read that's here, then I no longer can recognize this homology between two proteins that I know should be homologous, right? So this is the point that I'm trying to address. How do we identify things that are important without having homology to something that we know about, right? Because we don't know very much. And one way to do this is using embeddings, right? So we could ask the question, are these two embeddings similar, right? And for that, we would use cosine similarity. It's just a measure. I'm just going to skip that. But what we see very clearly is that these embeddings of these sequences are very different between homologous and non-homologous sequences, right? So in fact, simply by looking at these hyperdimensional embeddings of letters, without considering any homology, we're able to recognize homology versus no homology, which is a big deal, right? So it's kind of like you trained it to recognize the language, so you don't actually have to, point to objects for it to recognize that that's an object. In fact, when you look at the embedding similarity on the x-axis over here, versus the sequence similarity on the y-axis over here, you see that they're not very similar, right? So homologous sequences, which are highlighted in blue over the entire curve, over the entire graph, versus the non-homologous sequences distribute differently. But what we also see is that if we take the high embedding similarity, so anything that's over 0.6, we're able to capture homologous sequences, so the dark blue over here, even if sequence similarity is lacking, right? So if sequence similarity is below 50 bits, we usually don't consider that to be homologous. But if they fall into this group over here, we're able to recognize them as being homologous on the basis of the embedding similarity. And in fact, if I drew a line at sequence similarity of 20 bits, I would exclude all of these non-homologous sequences below the 20, right? So I could have a very high precision recognition of homology, even though I have no sequence similarity or very limited sequence similarity. And that's a very important finding. So if I look at evolutionary relationships, I do better if sequences are closer. So I do better for the same genus organisms, but I don't do that much better, right? So the difference is between 65% and maybe 80% if I'm looking at phylum level. But this is still above random, way above random if you compare sequences that you've not seen before. So the one thing that we saw, for instance, is if I look at the embedding similarities of the individual microbiomes, as you see, one from everything that's in pink is microbial biofilm over here, and everything that's in purple is host associated. And you see that microbial biofilm is very different from host associated samples, right? And stuff that's in green is water versus stuff that's in orange is in soil, and that's also very different, right? So these microbiomes are extremely different. And that's a very cool finding without ever using anything that we usually use in making these predictions. So one thing that we could do is, rather than trying to predict Mi-faser annotation, we can predict oxidoreductase functionality. And then we are able to predict significantly more. So this is Mi-faser with its 2% of predictions. And this is what our oxidoreductase functionality predictor can do 100%. And that means that it could recognize a whole bunch of sequences that we would otherwise not see. Now, of course, you can argue that you can annotate the metagenome just with random stuff. And in fact, you can. But if you look at the actual things that you expect to see, so at the surface, versus the halfway through the water column versus to the oxygen minimum zone, we expect a correlation with oxidoreductase activity. In fact, we see a reverse correlation for both MGREST and Mi-faser. For the EC classifier, for the oxidoreductase classifier from our deep learning, we see the correlation that we expect to see. So, some conclusions. Metagenomic functional differences across samples are important, right? As I showed you, even on 2% data. Mi-faser and soon LookingGlass models can highlight conditionally important functionalities. And the LookingGlass model can be used to answer any number of questions, right? So we can discover new functions, but we need to figure out how to validate the predictions. Now, very quickly, I'm going to mention FusionDB. This is just one of our ways of trying to bring up more functionality. So if you have individual microbes, we can annotate their microbiota. We can make predictions of metal binding for individual proteins, which is actually also well distributed across the microbiomes. So that's important. And that's about it. This is the people who did the work. I don't do the work, I just talk about it. And one of the things that I want to mention is that we're looking for PhD students. And if any of you are at the higher level, we're also looking for postdocs. And thank you.
Thank you, Professor Bromberg. It was an amazing lecture and a completely fresh look. I personally haven't had contact with it, and it was so amazing to learn something new from you. If anybody has questions, you can write them in chat. I can read them out loud for you, or you can just tune in and ask your question.
Thank you, Sophie.
It seems like you were so clear and great.
Or I lost everybody. One of those two.
I don't think so. It was pretty great, in my opinion. Okay, if nobody has any questions, we can conclude the meeting. And thanks for everyone who tuned in. And thank you again for your time and sharing your knowledge and expertise with us. We hope to cooperate soon again.
Well, we are looking for PhD students, so think about it.
Oh yeah. Well, I still have time until then, but this is definitely something to consider.
All right. Enjoy the rest of your day. Thank you for tuning in.